Hello students! Today we're going to explore some microscopic life. What would I use to explore microscopic life? Would I use getting really close to it? Would I use a magnifying glass? No, I would use a microscope. The microscope goes back many, many years. This is an example of an old microscope I have from about maybe the 1950s. This would be a field microscope you'd take out with you when you're going to explore things. And it's got a series of lenses in it. So a lens is a curved piece of glass, just like we have in this magnifying glass. Up at the top, you have a lens right here. We've got a lens at the top. We unscrew again, we have another lens. So the first lens magnifies it a little. The next lens magnifies it a bit more. And then down here at the bottom, we have the final lens, the objective lens. And this lens will do the final level of magnification. What you're going to look at would sit on a glass plate right here. And down here at the bottom would be a mirror. The mirror would be reflecting either light from the sun, or if you were inside, you might be using light from a candle or some electric light, but you're reflecting it up into here onto the glass plate, so you can observe what you're studying. The invention of the microscope allowed people to see things that were smaller than what could be seen with the human eye. It allowed them to investigate and find evidence for mysteries of life. Two examples of this are understanding the structure of what all life is made out of. For example, the cell theory that tells us that all living things, the basic unit of life for them is the cell. Here we're going to take a sample from a stepping stone where some water has accumulated. A lot of times mosquitoes like to lay their eggs in here, so maybe we'll find something mosquito related. Now let's look at some animal cells. How about mine? Alright, so I'm going to take a toothpick and I'm going to scrape some cheek cells off the inside of my mouth. I'm going to be very careful with this. I'm not trying to stab, I'm not trying to jab, I'm not trying to draw blood. I'm just scraping. So I'm going to do about 30 scrapes just to get a good amount of cheek cells. Alright, so now I have these on the end of the toothpick, and I'm going to smear them onto the slide. So I'm going to smear them, smear them, smear them. Make sure that it's all, and it's not super slobbery either. It's just the cells that got scraped off of my cheek. Put the slide on the stage. Turn on the microscope. I'm start with it on the lowest magnification, and I'm going to attach my camera to the microscope so that I can take good pictures for you. So I move around until I find something that looks like a skin cell, and at first it looks just like a smudge, but that's okay. So it doesn't look like a lot when you first look at it without any stain. That's why sometimes you add stain to it. So I'm going to add a little stain to it. First I'm going to put some red stain on it, and you want to be careful with stain because if you get it on your clothes, it could stain. Alright, so after sitting for a minute, I went ahead and rinsed it off, and you can see that there's some stained. Now I'm going to put blue dye on it. So now that it's stained, I'm going to look at it a little bit closer. So here is a chunk of skin cells. They're going to be blue and pink and purple. I'll zoom in a little closer, and you notice if you can see the tiny little blue dots, those are all the nucleuses, the nuclei of the cells. Go a little closer, there they are, skin cells from my cheek. The other thing that it helped us to investigate was number 2 is disease, sickness, illness. What is it that's going on that we can't see that's affecting our bodies? This allows us to understand the impact of microbes, things that are smaller than what we can see with the human eye. This could be protozoas, this could be bacteria, this could be viruses, this could be things that are
tiny, tiny, tiny.
In 1665, Robert Hooke looked through a microscope and he saw pieces of cork, a thinly sliced piece of cork, and he saw tiny structures reminded him of the places that monks would live, and he called them cells. He discovered the structure of living things. In 1674, Antony von Leeuwenhoek saw moving things moving around inside those cells, things that were cells themselves that were moving around, and he called them animalcules. And we have different names for them now because we know they're not all the same species and the same type of life, but we discovered that there was life, living, moving, active things at the cellular level. Let's talk for a minute about the history of medicine. It's embarrassing. In the 1840s in London, they kept having diarrhea outbreaks. There was a disease called cholera, and this caused you to have massive diarrhea, and everyone in your household would get it. It was horrible. It would destroy entire households overnight. The medical wisdom of the time was something called miasma. It was a matter of if you have bad air and something smells bad, it must be something that's ill and it's going to make you sick. It seems reasonable. Bad smells, right? Let's get out of here before we get sick. Clean air that smells good must be healthy, right? It seems reasonable. We weren't even worried about dirty hands or dirty dishes. Just wipe them off and go on about your business.
What's the harm?
As long as they weren't too smelly. Doctors would perform surgeries and washing hands wasn't even part of the procedure. They might rinse off some instruments, wipe off the gunk, but they weren't worried about cleanliness. They weren't worried about cleaning anything. They had no idea about germs. They had no idea about microorganisms. Really, smell was the thing that meant the most to them. In the 1840s, a lot of this was about to change. John Snow was a man who started to investigate the cholera outbreak in London in the 1840s. So in the 1830s they started having it, 1840s they're having it. They had cholera outbreaks into the 1850s in London, and it was the strangest thing. You'd have one household that is entirely healthy, and the next household over, it'd be everybody dying. Rich homes and poor homes, clean homes, dirty homes, spaced apart. It's really crowded, and they would both get sick. People couldn't figure out, why are we getting sick? You'd have rich households, poor households, clean households, dirty households. What was going on to make everybody so sick? Snow figured out that all of the households that were getting sick were getting their water from the same well. The same source of water was causing them to get sick, which meant there was something contaminating that water. He didn't know what. He had no idea about germs at the time. He had no idea what it was that was causing them to get sick, but he had to work really hard. Eventually he was able to convince the scientific community that it was the water from the well and they needed to stop drinking that well water. In Austria, at the same time, there was Ignaz Semmelweis. He was in a hospital that had two different wards of women. They were having babies, and one room had a whole bunch of women having babies and they were just fine. Another room had a whole bunch of other women that were delivering babies and they were dying left and right. They were getting something called puerperal fever, and it was something that they would get after delivering the babies. Nobody could figure out what was going on. So he investigated. He tried some different things. He said, well, what if we stop ringing bells around them? What if we have them lay on their backs instead of their sides? What is it that would be the solution? He finally figured out that the hospital ward where all the women were dying was the one where the medical students were delivering the babies. Now, it wasn't because they were medical students. It was because they were performing autopsies. They were cutting open dead bodies before going to deliver babies. That means they had chunks of dead bodies on their fingers. And while they would brush them off a little bit, they still had bits of dead flesh on their fingers. And that had contamination that was getting the women sick. So he said, all right, you guys have to start washing your hands. I don't know why, I don't know how this is happening. Again, he had no idea about the microbes yet. He knew that there was something about what was on their fingers that was getting the women sick. So he started having them wash in a chlorine solution, not even soap. He went straight to chlorine, not because it would kill the bacteria, but because it was something that would get rid of the smell, and it worked. Why people were getting sick was still mysterious, but we were getting closer to the answer, understanding there was something that we could not see with our eyes that was getting people sick. Then in 1854, in Italy, a man named Pacini was able to look at a microscope and he actually saw the cholera bacteria that was getting everyone sick. Now, cholera bacteria is very, very small. A skin cell, now here's an example of a skin cell. A skin cell is about 35 micrometers long. Cholera bacteria is about 1.5 micrometers long. the cholera bacteria that was getting everybody sick. So it is really, really, really tiny. So you would need a really good microscope to see that sort of thing. The advent of the microscope led to being able to identify diseases, not by looking at the symptoms, not by smelling the air, but by actually looking at the microorganism that was causing the disease. Now, the idea of germs causing diseases was something that took many years before it was widely accepted. We're still learning more about bacteria and viruses and prions and things that we can't see that are causing a disease. But it is on the path to learning more about how to keep ourselves healthy.